Welcome back to ZOO3649 Evolutionary Genetics. This is lecture 30, the last lecture in the section Genetic Structure. And genetic structure is the fourth section out of 6 sections in ZOO3649. So, we have been looking in this section, genetic structure, about characters, traits, and which traits, homologous traits, that can be used to make what? To make trees. Okay? The idea of trees comes from Darwin. Okay? And the idea of trees is to try to compartmentalize or to divide up the variation in life that we see around us into into groups where we can trace back the lines of evolutionary descent. That is why tree building is so important in evolutionary genetics and in all of biology. Okay? Because we need to be able to know the relationships between the different species we have. Okay? And so building trees is one way to figure out those relationships, those genealogical relationships. Okay? That's why tree building is so important. On the other hand, in the previous section, section 3, population genetics, we talked about populations. And we talked about what? We talked about alleles, frequencies, right? Big frequency of big A and small a. And what happens when we have different forces of evolution? Remember in our practicals, right? So what happens when we have a small population size? Genetic drift will push alleles to fixation, right? What happens when we have selection for heterozygotes? Then balancing selection will keep two alleles in the population. And if the population size is large enough, it will overpower genetic drift. If the population size becomes too small, genetic drift overpowers balancing selection. So you know about all these population level things and how allele frequencies change from one generation to the next. And that is how we even measure evolution, right? So the idea in this lecture is, and this is what we are using now in modern genetics, we're using coalescent methods. We're not using tree methods. We're not using population methods. We're using a combination of both methods. And this combination is called the coalescent framework. Okay? So the coalescent framework marries together the idea of populations, what we learned in section 3, and the idea of trees, which we learned in this section. And that's why the coalescent is so powerful, because what? It brings together the populations together with trees into one holistic analysis of evolution. And that is why the coalescent is so powerful in evolutionary biology. All right? And so we're going to talk about the coalescent today. So if you recall the Wright-Fisher model, right? Remember, we talked about extensively about the Wright-Fisher population. What is a Wright-Fisher population? It is a population an idealized population that was developed by Sewell Wright and Ron Fisher independently to model genetic drift. Why? Because the Wright-Fisher population controls for all other forces of evolution except for genetic drift. Because the Wright-Fisher population always has a constant population size. Right? As soon as the population size is infinite, you are not modeling drift. Okay? Because then you have Hardy-Weinberg equilibrium. All right? So the Wright-Fisher is the next step from Hardy-Weinberg, right? Because it is not saying, well, we have a change in allele frequencies, but we don't know what force is creating it. The Wright-Fisher tries to say, well, we have a change in allele frequencies, and it is due to genetic drift, or it is not due to genetic drift. Okay, that's what the Wright-Fisher population can tell us, because the Wright-Fisher population is a constant size. So recall the Wright-Fisher model, the Wright-Fisher population, which we learned about. This is what we call a forward projection model. Okay, now what I mean by that is, imagine here's your Wright-Fisher population, okay, and you start at the top line. So the top line is generation 1, okay, and so you see there that there are lines in the top from the top dots. The top dots is each individual in the generation 1, and you see the lines connecting the individuals in generation 1 to where? The individuals in the next generation, okay? So those lines that connect the dot in the top line to the dots in the second line, those are the parents of the dots in the second line. Okay, and you can trace the lines of descent or lines of ascent. Okay, so this is starting here and moving forward in time, and you can see based on these lines which individuals in each generation gave rise to individuals in the next generation. So here are the ancestors, and here are the descendants. Okay, so you start up here, and the genealogical process then moves forward with time. Okay, and you see where one generation leads to the next generation, and leads to the next generation, and leads to the next generation. Okay, so we're going forward in time with the Wright-Fisher population. And remember also that the Wright-Fisher population has a fixed population size. The population size here is the width. So it's 1, 2, 3, and it's a fixed size. So this number here doesn't change from generation to generation. Why? Because we want to model genetic drift. Okay, so what's the problem with the Wright-Fisher population? What's the problem with modeling something going forwards? Okay, what is the problem? The problem is that if you do that, then much of the genealogy in the forward direction is not relevant. Why? Because not all the individual in this generation will have kids, right? So if you have to start with generation 1 and go forward in time to generation 2, you are going to have to model the probability of every individual in generation 1 having an offspring in generation 2. It could have an offspring, but it might not have an offspring in generation 2. So the problem is that if you don't have a offspring in generation 2, we spend a lot of time modeling that outcome, but it's irrelevant to model that outcome. Okay? Because a particular individual didn't actually reproduce in generation 2. But we don't know that because what? We're trying to model in the forward direction. Okay? So in other words, look at this. This is trying to model, okay, the Wright-Fisher population in a forward genealogy. You have to then model every single individual in every single generation contributing to the next generation. Okay? But as you see here, for example, this individual here, and that individual there, and that individual there, you see where my mouse is moving? Those individuals didn't have offspring. But you spend a lot of time, computerized time, to model whether they will have offspring or not. But they didn't have offspring. So you spend a lot of time modeling irrelevant stuff. Okay? Because if you actually want to see what the important part of this Wright-Fisher population is, what's the important part of this whole Wright-Fisher population? It's not everything that you see here. It's not everything. It's this. This is the only important. This is the very same population, but it's showing only the important lineages of descent. Okay? So this one, if you start here at the last generation and work backwards, then you get this backward genealogy. And you trace all the today's individuals, all of them, you can trace them back using their lines of ancestry and descent to a common ancestor, the most recent common ancestor, what we call the MRCA, most recent common ancestor of this Wright-Fisher population. Okay, so basically you see the difference between these two figures here. Here's the forward genealogy, here's the backward genealogy. Which one involves more calculations? Obvious, the forward one. That's what the forward one involves more calculations. And that is why I say much of the genealogy going forward is irrelevant. Because why? Only this part of the genealogy is relevant. Because only this part shows an unbroken line of descent from an ancestor to all the current individuals of the population. Okay? This one shows it has that information, but it has a lot of other nonsense information. For example, all these guys here, like here, these guys here, and here, and here, and here, that what? That didn't have an offspring. But nevertheless, we had to calculate that. So let's stop with going forward and all the useless, irrelevant calculations. Let's start with what we have today and work backwards. And then we have fewer calculations to do. So you see now why going backward, okay? Why not start here at the end and work backwards towards the ancestor? Because that way you are then dealing with fewer calculations, okay? And not so much calculations of irrelevant stuff. Okay, so it makes more sense to go backwards. And that is what the coalescent is all about. The coalescent is about going backwards in time. Okay? That's kind of what coalescence even means, right? Because if you think about what coalescence means, when something coalesces, it's like when two lines that are going in different directions meet. Okay? For example, when you are driving on a road and the road forks in two different directions, right? If you're going forward in time, you can go in this direction, right? Or you can go left, right? But if you're going backwards in time, so you will see, if you're going forward, you will see the road diverging, okay? But if you are coming backwards in time, what would you see? You would see the road becoming one. You would see two roads becoming one road if you're going backwards in time. Okay? In other words, you will see the two roads coalescing into one road. Okay? And that's what we mean by coalescence. So we start with today, we go backwards in time, and we see where all the DNA sequences and alleles and all the genetic information coalesces towards a single ancestor. That is the power of coalescence. All right? So you don't have to, and here you have again the picture of our right fisher population here with where my mouse is, right? This is the right fisher population. If we started here and went forward in time, we would have to calculate every single in other words, every round dot here, we would have to model. It would take an enormous amount of computer time, okay? But if we don't do that, if we start here and we don't even sample every member of the population. We only sample these three in blue. Even if we don't get all those guys sampled, we don't take all six, we take only these three, we can still figure out the important part of the tree, of the right fisher model is going where? The important part is this blue part. Because that's the only line that leads to the ancestor. All this other stuff in white, they are the irrelevant, useless calculations. Okay? So it's this blue part of the right fisher population that we're really interested in. Okay? And you see how few calculations you have to do going backwards. Compared to if you're going forward, you have to do every one of these calculations. Okay? So we start with what we know, that is here, where the arrows are, the DNA sequences of individuals that you have sequenced today, in the present, at time 0. And then we simply use the right fisher model in reverse. We put the right fisher into reverse gear. Like you're driving a car, going forward? No, you put the right fisher into reverse and you go backwards and you work out how long, you work out what was the ancestral DNA sequence and how long ago in time, in generations, did it exist. Okay, when was the common ancestor alive? How long ago did the common ancestor live? Okay, this really simplifies things because now we don't have to worry about the entire population, this entire population here. We simply have to deal with the histories of the individuals that we have sampled. And those 3 individuals that we have sampled will give you the true history going backwards to the ancestral DNA sequence. Okay? But of course, a simple tree has its limitations. Okay? There is not much information about a right fisher population processes happening as the population is evolving. Okay? By when I mean By a simple tree, I mean the trees that we've been building for the last 5 lectures. Remember, we've been talking about tree building, right? Substitution models, likelihood, Bayesian, distance, parsimony. These are phylogenetic methods to build trees. They also work backwards in time, yes. But those making trees, the way we have been talking about all these lectures, has a limitation because why? The tree does not include population information. Okay? The tree, when you make a tree, does not include these Wright-Fisher processes. So by not including population information, you don't know about the rate of allele frequency changes in the population. And remember, that's how we measure evolution. We measure evolution by allele frequency changes from one generation to the next generation. Okay? There's no information about the mutation rates, about the population sizes, and there's no information about migration and selection as you go backwards in time when you make a simple tree, the way we've been discussing for all these lectures. All right? So a simple phylogenetic tree using likelihood or Bayesian has its limitations, okay? Because the genealogical process that it recreates is independent of the mutational process. But we want to bring both the genealogical branching process of trees together with the mutational process and the allele frequencies of populations. We want to bring these two great ideas together into one framework. Okay? So coalescence, the coalescent framework, is what brings these two great ideas. The idea of trees, which we've been discussing for these last lectures, and the idea of populations, which we discussed in the 3rd section of the module. These two ideas, the tree idea and the population idea, we want to bring them together. Okay? And we want to synthesize this into a framework, and this is what coalescence does. Okay? Is where population thinking about allele frequencies meets tree thinking about branches of descent, working backwards. Okay? So coalescent theory is a study of the statistical properties of the gene genealogies under different assumptions. Okay? And the main idea is that a gene genealogy, a tree, underlies the data that we have sequenced. Okay? The main idea is that we sequence genes in many different individuals, and a gene genealogy is what links all these sequences together going backwards in time. All right? So by integrating the population approach, which deals with what alleles and allele frequencies and the evolutionary forces that are changing them, with the genealogical approach, which says that evolution is tree-like, bifurcating trees, we end up with what we call coalescent theory. And this coalescent process is a much, much more powerful tool for modeling the population genetics, the genetics of a population, than traditional population or traditional genealogical methods. Okay? So by traditional population methods, I mean looking at a genealogical, looking at a Wright-Fisher population going forward. Okay, that's a traditional population method. And the genealogical methods I'm talking about are the tree building methods that we talked about in the last lectures. Okay, they're both are excellent methods, but we want to combine the two excellent methods to give you coalescence, which is an even better method than both of them by themselves. Okay, so the basic coalescent, let's go through this, but let's try to understand coalescent, the basics of coalescent theory. And I'm teaching you this because, yes, it is going to be stuff that you need in your assessments, but it's also stuff if you're thinking of doing honors next year in zoology. My dear friends, if you do not understand these basic theories, you will be lost and you will find it very difficult to pass the honors module, molecular ecology, which a lot of students fail every single year at honors. And you don't want to be one of those students that has to come back again and do honors for a second year. Do you? No, you don't. So please It's all about understanding. You don't want to be caught lacking basic information in honors, do you? Okay, so the basic coalescent I'm going to explain to you. Imagine we have a right Fisher population. We have these dots here in generation. These dots are either, they can be individual people, they can be alleles, they can be a DNA sequence, okay, they can be any anything that you have sampled, yeah, in one generation. And generation 2 is then the offspring of generation 1. Okay, with time going from generation 1 backwards. Okay, so in other words, we are looking here at the present and we're going backwards. We're looking at the ancestors. So if this is generation present, this will be parents, obviously, right? And if there's another line up here, it will be grandparents. And the next line will be great-grandparents, and so on. We're going backwards in time, remember. Okay, and the population is constant in size. So the number of individuals in one generation is equal to the number of individuals in another generation, okay? So in this system, we've got two alleles, all right? You can call them big A, small a. You can call them, in this case, we have the yellow allele and we have the blue allele. And you see the yellow allele is not so frequent, whereas the blue allele is very common. So the first step, okay, is to figure out what is the probability that any one allele, either yellow or blue, in the second generation, so in the offspring's generation, okay, in the parent's generation, okay, has a parent in generation 1. What is the probability that an allele in this generation here has a parent in this generation? What is the probability going backwards? The probability is that obviously it has to be 1, right? Because if an allele appears in generation 2, where does it come from? It obviously came from generation 1, okay? So if you have an allele in this generation, right, the probability that it, the other way around. If you have an allele in this generation, the probability that it came from a parent in the preceding generation is equal to 1, okay? The probability of having a parent is equal to 1 because everybody has a parent, okay? You come from somewhere. Nobody just sprang out of a hole in the ground, right? Everybody has a parent. So the probability of having a parent in the previous generation is always 1, right? Okay, so then, now that we got that, how about what is the probability that 2 alleles in this generation, generation 2, are coming from the same parent? Okay, the first question was, what's the probability that 1 allele comes from a parent in generation 1? That probability is 1. Now the question is, what's the probability that 2 individuals with the same allele have the same parent in the previous generation? And that probability will be 1 divided by 2n, since what? The chance is dependent on the size of the population, right? The probability of coalescence between one generation and the next is 1 over 2n. Okay, and remember, now that you see that, why is it 1 over 2n? Because you have to divide by the size of the population, and 2n is the diploid. We have a diploid. Remember, right Fisher is a diploid population, so it's always 2n, okay? So it's 1 divided, the chance is 1 divided by 2n, that 2 individuals in generation 2 have a parent in generation 1. Okay, so that is the whole essence of coalescence. And remember, this is also a way to model genetic drift in a right Fisher population. Remember We we have discussed how to model genetic drift in a right fisher population. You have the divergence at 1 over 2n between two right fisher populations. You have the increase in homozygosity at the rate of 1 over 2n, the inbreeding coefficient. And this is the last way to do it, using the coalescent. The probability that two alleles or two individuals in generation 2 have the same ancestor in generation 1 is 1 over 2n. It's the same formula, isn't it, that we used to model genetic drift in the other two examples. It's always 1 over 2n, because that is the golden rule of genetic drift in right fisher populations. Okay? So there are three ways to model genetic drift in a right fisher population. If you ask this, and that's why I say, if you're asked an exam, like in an exam or in a test, a question like that, how do you model genetic drift in a right fisher population? Okay? If you memorized just from the right fisher population, if you just memorized that lecture and you thought you know the answer, you don't know the full answer, because you will not get the full marks, right? Because the true answer, you have to be thinking, and you have to realize, Moodley has given me the true answer, but not in one slide and not in one lecture. I have to find the true answer here, there, and everywhere in the module. And because I am understanding the module, aren't I? If I'm understanding the module, I will realize 1 over 2n. Where have I seen it before? Oh, in the coalescent process. Oh, in modeling genetic drift in right fisher. And I will be able to put them all together in one answer when I'm writing my exam or when I'm writing my test. Okay, so that you can't memorize one slide and expect you know the right answers. You might pass if you're lucky, but you won't get a distinction. Okay, you're never getting a distinction from memorizing, and that means you're not going to do an honors in genetics. All right, so please understand. This is how we model genetic drift in a right fisher population. 1 over 2n using this coalescent process. And why 1 over 2n? Because the probability of coalescing of two individuals in generation 2 coalescing to the same parent in generation 1 is 1 over 2n. It's depending on the population size. Okay? So it's a function of genetic drift, because genetic drift is also depending on the population size. So then obviously, if the probability of coalescing if two individuals coalescing in the previous generation is 1 over 2n, then what is the probability of not coalescing in the previous generation? Obvious, right? It's 1 minus 1 over 2n. Okay? That's the probability of not coalescing. It's also dependent, of course, on the population size. And so that way, we can figure out the probability going back many generations. Okay? Because each generation where two individuals do not coalesce, it's simply 1 minus 1 over 2n. Okay? Until you get to the generation where it's 1 over 2n and they do coalesce. Okay? And you work backwards in time, generation after generation, until you find that place where all the alleles coalesce to a common ancestor, the most recent TMRCA, the most recent common ancestor. Okay? So, if we go back then another generation, the probability of the two alleles coalescing 2 generations ago is 1 minus 1 over 2n, which is the probability of not coalescing in one generation, multiplied by the probability of coalescing in the second generation. Okay? So then if you had to go, what is the probability that two alleles coalesce in 3 generations? If I asked you a question like that in the test, what would you say? It is 1 minus 1 over 2n times 1 minus 1 over 2n. That's for the not coalescing in 2 generations. And then lastly, in the 3rd generation of coalescence, times 1 over 2n. Okay, so the 1 minus 1 over 2n, you add another one and another one depending on how many generations you go. Okay, and it therefore follows this pattern here. It's this geometric distribution where you add simply, if you probability of coalescing 2 generations, you actually have 1 minus 1 over 2n, right? And 3 generations, you basically have 1 minus 1 over 2n squared. In other words, multiplied by itself again. Okay? And so with the number of generations increasing, okay, number of generations to coalescence increasing, you simply add another 1 minus 1 over 2n at each point, okay, that it's not coalescing, until the coalescence generation, which is always the last one, 1 over 2n, okay? And so this is the geometric distribution with a mean of 2n for the coalescent, using with 2 gene copies. In other words, where there are 2 alleles in the system, a big A and a small a, say, or a yellow and a blue, like in this case. But we know, okay, in the real world, there are many more than just big A and small a in the population. There are many alleles. Some loci have hundreds of alleles, like microsatellite loci. So what happens when there's more than 2 alleles in the population? And a very clever guy named Kingman in 1982, he came up with a formula, this formula here, when there are k alleles in the population. In other words, when there is more than 1 allele, more than 2 alleles in the population, okay? So k is basically equal to the number of alleles in the population. So if we have a population with 10 alleles, then k in this formula will be 10, and you will have 10 times 1 minus 10, which is 9, divided by 2, okay? And that basically gives you the number of pairs of gene copies in the Wright-Fisher population, okay? So the number of gene copies is simply the number of alleles multiplied by 1 minus the number of alleles and then divided by 2, okay? Because, as you know, it is always a diploid population that we are dealing with in a Wright-Fisher population, okay? And so then once you've got that probability, once you know the number of gene copies, then you multiply it simply by the probability that a pair is coalescing in the next generation, okay? So if you take this formula, multiply here these 2 terms, what happens? You actually get k brackets k minus 1. You multiply by 1 here, nothing changes in the numerator, but what happens in the denominator? This 2 times 2 gives you 4, and you end up with this formula here, okay? And so this is Kingman's famous formula for the probability of coalescence in a Wright-Fisher system going backwards, in a coalescent Wright-Fisher system going backwards in time, with k alleles in the population, okay? And it's called Kingman's N coalescent. So how do we use coalescence to estimate the population parameters going backwards in time? Okay, the population parameter theta can be derived from coalescent theory. I'm not going to show you how we derive it, that is for honors, but suffice to say that we can work out theta, which is equal to 4 times the effective population size, which is the way to measure drift, times mu, which is what? That little u with a squiggly bit. What is that? That is the mutation rate. Okay? So the Wright-Fisher population, as you know, it's this coalescent theory is always taking into account the effective population size and the mutation rate. And the product of that multiplied by 4 is equal to theta, which is the coalescent population parameter. Okay, and so if you know theta, then you can estimate NE, right? You can derive NE because you just put 4 and the mutation rate on this side divided by theta, and you got what? You got the NE. Or if you got the NE but you don't know the mutation rate, then you can put 4 and NE on this side and divided by theta, and you got what? You've got the mutation rate. So the coalescence allows you to calculate population parameters based on allele frequencies going backwards in time. So you see, you're getting the best of both worlds. You are looking backwards in time like a tree where lineages coalesce, branches coalesce, but you're also getting population level data, population level results and information at each step going backwards in time. Okay? So you got the population stuff, the population information, and you got the tree information together. And that's what coalescence brings to the table. Okay? And so you can extend this idea of coalescence to include more than one population. Here's a coalescent theory. So here's a coalescent. Here you can see now 1, 2, 3, 4 different right Fisher populations going backwards in time. And we know they're right Fisher populations because it's not just one line, okay, like a tree. It's a population. Here are 5 individuals in this population. Here's another population going backwards in time. Okay, so it's all populations, right Fisher populations going backwards in time. And overall, they will show you the overall branching patterns of branching descent, which is this overall tree-like pattern you see here. But it's not just a tree, is it? It's populations. Inside each branch of the tree, you have the population as well. So you bring the population of the right Fisher population and you bring the trees together. Okay? And you marry them into one holistic coalescent analysis. Okay? Additionally, what you can do is you can model migration between the populations. So you can model migration from this population here where my mouse is to this population, the purple one, or from this orange ancestral population to the purple one, or from this purple one to one of these derived populations here that you can actually then add selection, recombination. In fact, you can make the coalescent model as complicated as you want to make it, okay? But, like with all complex problems in biology, what happens? We run into the problem of parameter space becoming huge. The same problem we had with our Bayesian and likelihood tree inference. The same problem we get with coalescence, because we are also trying to use a likelihood or a Bayesian approach to figure all this out, to figure out the population processes as well as the tree processes. Okay? So we are using both sets of information. Okay? So we also need a likelihood and Bayesian way to do coalescence. Okay? And what happens when you have these complex models? You need to find a Markov chain because it is impossible to search the whole parameter space to find the right genealogy and the right population parameters. Okay? Because now your coalescent model is much more complicated than just a simple tree. Okay? Because a simple tree doesn't have the population information. Right? Now you've got the tree, you will need to estimate, which already required a Markov chain and Bayesian and likelihood. Now you've got the addition of populations on top of that. And so you will always need a Bayesian or a likelihood approach, including a Markov chain Monte Carlo sampling, to work out the coalescent model, to get to the bottom of what is the genealogy, what is the coalescent genealogy, and what are the population parameters going back at each generation, going backwards in time. Okay, so the coalescent is very, very powerful. It marries the two big ideas, and this could well be a test or exam question, guys. Why is coalescence such a powerful tool in evolutionary biology? The answer is what? The answer is because it marries the two great thoughts of population genetics, the modern synthesis, and the idea of making trees. It marries the two of them into a powerful framework of going and of looking backwards in time, retracing the lines of descent of the tree, but at the same time working out the population parameters going backwards in time. Okay? And that is why the coalescent approach is the best approach we have in genetics at the moment. Okay, with that, I would like to leave the section of genetic structure. We are done now with genetic structure, and in the next lecture, we will start with our new section on speciation, section 5 of the module ZOO3649, Evolutionary Genetics. Okay, so let me stop the recording and I will see you for the next lecture.